12 Biology, CBSE Term 2 syllabus and mark allotment. Theory part, chapters and units for Term 2 CBSE board exam. Unit 8, 9 and 10. Unit 8, Biology and Human Welfare. Chapter 8, Human Health and Disease. Chapter 10, Microbes in Human Welfare. Learn Chapter 8, Human Health and Diseases without much effort. Let us study human health and diseases. A revision. Health is a state of complete physical and mental and social well-being, and not merely the absence of disease or infirmity. Factors affecting health. Genetic factors. Example, sickle cell anemia. Lifestyle. Example, obesity. Infections. Example, influenza. Types of diseases. Infectious diseases and non-infectious diseases. Infectious diseases can be called as communicable diseases. Example, leprosy, common cold. They are transmitted by air, water, physical contact, and animals. The causative agents may be viral, bacterial, fungal, worms, and protozoans. Control methods: balanced diet, good personal hygiene, awareness about diseases, vaccination, hygienic food and water. Non-infectious diseases can be called as non-communicable disease. Example, high blood pressure, cancer, heart diseases. Causes, stress, no or less physical activity. Genetic, unsuitable diet, wrong lifestyle, exposure to carcinogens. Control measures, stress management, physical exercise, balanced diet, avoiding drugs, alcohol, and tobacco. The diseases are of two kinds. They are contagious and non-contagious. Now let us see about various types of diseases. First one, diseases caused by worms. Ascariasis. The pathogen is roundworm, Ascaris lumbricoides. It spreads through water, vegetables, fruits that are contaminated by feces of infected person, or simply we say unwashed vegetables and fruits. Symptoms of ascariasis: internal bleeding, muscular pain, fever, anemia, blockage of intestinal passage. Elephantiasis or filariasis. The pathogen is Wucheria. There are two types of Wucheria, Wucheria malai and Wucheria bronchiopti. They spreads through bite of female mosquito vector. The symptoms: chronic inflammation of the organs, usually the lymphatic vessels of the lower limb. Is malaria. It is caused by Plasmodium. Let us see the life cycle of Plasmodium in a short. Plasmodium requires two hosts to complete its life cycle. One is vertebrate, the human being. The other is invertebrate, the mosquito. The infected female Anopheles mosquito consists of sporozoite. When bites a healthy person, it releases Plasmodium sporozoite into the blood of the human. These parasites multiply in the liver cells and finally burst the liver cells, and sporozoites are released into the blood. These parasites enter RBCs and further multiply here. And finally, burst RBCs also. Bursting of RBCs is accompanied by a release of toxic substance called hemozoin, which is responsible for fever and chillness. In the RBCs, only sporozoites change into gametocytes. Gametocytes multiply. When the deceased person is bitten up by a female Anopheles mosquito, gametocytes are introduced into the mosquito due to sucking of blood from the infected person. Gametocytes fertilize and develop inside the intestine of mosquito to form sporozoites. These sporozoites are stored in the salivary gland of mosquito and are released into the healthy person who is bitten up by this mosquito. We saw some of the diseases that affects human being. Though there are many diseases that can affect human being, our body has some special power to protect us from various infections and pathogens. This power we call it as immunity. So let us see about immunity. What is immunity? The ability of the body to fight against the disease causing organisms and protection is called as immunity. Types of immunity, and the immunity is of two types. They are innate immunity and acquired immunity. Innate immunity, it is present from the time of birth. It is non-specific. It consists of four kinds of barrier. They are physical barrier, physiological barrier, cellular barrier, and cytokine barrier. First one, physical barrier. It is skin, mucus coating of respiratory, gastrointestinal, and urinogenital tract that prevents the entry of microbes into our body. Next is physiological barrier. They are acid in stomach, saliva in mouth, tears from eye. Cellular barrier. They are blood. Blood has leukocytes such as polymorphonuclear leukocytes, monocytes, etc. And tissues has macrophages which phagocytose the microbes. Next one is cytokine barrier. They are special proteins called as interferon and are secreted by virus infected cells that prevents the further spread of viral infection. The other is acquired immunity. It is acquired. It means that the immunity produced in the person in response to an encounter are faced are already affected with the pathogen by the individual earlier. It is in the memory of immune cells. Acquired immunity is pathogen specific, and the immunity or immune response is of two types. They are amnestic response and amnestic response. The amnestic response is if a pathogen for the first time infects a person, low intensity immune response is generated. We call this first time response is called as primary response. Next one is anamnestic response. If the same pathogen attacks again, heightened immune response is generated in the person, thereby preventing the occurrence of disease in a heightened immune response. In acquired immunity, two types of cells are involved. They are B lymphocytes and T lymphocytes. Let us see about the B lymphocytes first. B lymphocytes secrete a protein called as antibodies in response to the pathogen infection. Antibodies are specialized glycoproteins with four peptide chains. They are two light and two heavy chain. Hence, these chains are denoted as H2 and L2. H2 means two heavy chain, L2 means two light chains. The antibodies or immunoglobulins are five types. They are IgG, IgA, IgM, IgE, IgD are examples of some of the antibodies. They generate humoral immune response since they are present in the blood. T lymphocytes. T lymphocytes help B cells to produce antibodies. They generate cell mediated immune response. This response helps to differentiate self and non-self cells in our body, as in the case of graft rejection. You can understand this during graft rejection. During organ transplantation, the immune system of the recipient will reject the donor's organ. It is because of this response we call it as response to differentiate self and non-self cells of our body. The immunity can further be divided into two types: active and passive immunity. Active immunity. This is the naturally acquired immunity produced in the host body in response to an antigen. It is generated in our body by immunization, and by body naturally gets immunity to a microbe that had caused infection previously. Then we'll see about the passive immunity. When ready-made antibodies are provided to an individual to protect against foreign agents. Example, colostrum. Colostrum present in the mother's milk, and they contain IgA. Also, the fetus gets antibodies from mother through placenta. Vaccine. It is a substance used to stimulate the production of antibodies and provide immunity against one or several diseases. These vaccines are prepared from the causative agent of a disease or its products or a synthetic substitute. These are treated to act as an antigen without inducing the disease. How does the vaccine help? Vaccines are nothing but inactivated pathogens. These inactivated pathogens are vaccine, when introduced into the body of a person, produces a primary immune response, and antibodies are also produced against this pathogen, and they too develop memory B and T cells. Now, when the same pathogen attacks again the same person, memory B and T cells generate a huge and immediate immune response, and the pathogen is killed. Immune system normally gives us protection, but there are some diseases which are caused due to malfunctioning of the immune system. So now let us see some of the problems related with the immune system. The problems of immune system are allergies and some of the autoimmune diseases. Let us see about allergies first. Allergies, an exaggerated immune response to certain antigens present in environment, and the substance which cause allergies are called as allergens. Allergens, substance in response to which allergy is produced. Example, dust, pollen grain. Antibodies involved in allergies are IgE. During allergic reaction, chemical substances such as histamine and serotonin are released, due to which the following symptoms are produced. The symptoms like sneezing, watery eyes, difficulty in breathing, etc. may arise. The allergic test. Patient is injected with small doses of allergens to monitor his response to this allergen. Control and drug for allergies. They are antihistamine, adrenaline, and steroids may be given so that the symptoms of allergy subside or stops. In autoimmunity, the body generates immune response against its own body cells. Reasons may be genetic and other unknown reasons. Examples for autoimmunity: rheumatoid arthritis. The immunity of our body is provided by a system called as immune system. So let us see human immune system in a nutshell. Immunity provides us protection. This immunity is provided by immune system. An immune system gives us protection through lymphoidal organs. The lymphoidal organs are of two types. They are primary lymphoidal organ, secondary lymphoidal organ. Primary lymphoidal organs consist of bone marrow and thymus. Here, immature lymphocytes are differentiated to form antigen sensitive lymphocytes. First one, we'll see about bone marrow. Here, all blood cells including lymphocytes are produced. Then, thymus. It is responsible for maturation of T lymphocytes. Thymus, it is a bilobed organ which is situated near the heart and keeps on reducing in size as the age increases, specifically from the time of puberty. Then, secondary lymphoidal organ. Lymphocytes migrate here after attaining maturity. The secondary lymphoidal organs are spleen, lymph node, tonsil, Peyer's patches, and appendix. First, let us see about spleen. Spleen is the large bean-shaped organ containing lymphocytes and phagocytes, which acts as a filter for blood, and it filters the pathogens. Next, lymph node. They are located at different points throughout the immune system. They trap the antigens present in lymph or tissue fluid, and these antigens cause activation of lymphocytes and generation of immune response to themselves. Tonsil. Because of their location at the throat and palate, they can stop germs entering the body through the mouth or the nose. The tonsils also contain a lot of WBC, which are responsible for killing the germs. Next, malt, mucosa associated lymphoidal tissue. Malt lines major tracts and constitutes about 50% of lymphoidal tissue in our body. This malt or mucus, they present in respiratory, digestive and urinogenital tract. AIDS, acquired immunodeficiency syndrome, caused by HIV, human immunodeficiency virus. HIV is a retrovirus. Its genetic matter is RNA. So we call RNA virus. Transmission of HIV occurs through sexual contact with infected person, sharing of infected needles, transfusion of contaminated blood, infected mother to child through placenta. The time lag between infection and appearance of symptoms are few months to many years. How does AIDS infection spread in our body? Virus first enters into the body of a person and enters into macrophages. Here the virus replicates. How does this virus replicates inside the macrophages? The viral RNA reverse transcribes to viral DNA. The viral RNA reverse transcribes to viral DNA, which gets incorporated into host DNA and subsequently new viral particles are produced. Thus, macrophages become a viral HIV factory. Therefore, HIV enters into helper T lymphocytes. Here, they replicate and produces many progenies. As the progenies are released, they attack other new healthy T lymphocytes. Therefore, T lymphocytes start decreasing in number and immune response of the person becomes weak. Even infections which could be overcome easily start aggravating. Diagnostic methods of AIDS. AIDS can be diagnosed by ELISA, enzyme-linked immunosorbent assay. Treatment of AIDS. There is no permanent cure or therapy for AIDS, but antiretroviral or antiretroviral drugs can be given. Prevention of AIDS. Use of disposable syringes, screening blood and blood products from blood banks, advocating safer sex, and many organizations are giving awareness to the public. One is NACO, National AIDS Control Organization, and many NGOs are doing a lot to create awareness among people. The process of development of cancer is called as oncogenic transformation. Normal cells have the property of contact inhibition, but cancer cells lose this property. What is contact inhibition? Normal cells will stop when they touch the nearby cell during their growth, but cancer cells do not stop when they touch the nearby cell, so they keep on growing. So cancer is nothing but uncontrolled growth of. As a result, cancer cells divide continuously to give rise to mass of cells tumor. Tumors are of two types. They are benign tumor and malignant tumor. First, we will see benign tumor. Benign tumor remain confined to the original location and do not spread. And second one is malignant tumor. These malignant tumors exhibit metastasis and they reach distant places, and wherever they reach, they lodged there and new tumor is formed at the site where they have been lodged. So malignant tumors are actually represent cancer. The malignant cells actively divide, grow and starve the normal cells of vital nutrients. Causes of cancer. The cancer causing agents can be called as carcinogens. The physical agents are ionizing radiations and non-ionizing radiations. Ionizing radiations are X-ray and gamma ray. Non-ionizing radiations are UV. Biological agents are viruses. Biological agents like viruses will have viral oncogenes. These oncogenes are cancer causing genes, self genes. Sometimes normal genes in our body cells called proto-oncogenes get converted into cellular oncogenes that can cause cancer. Normally, our body cells have these proto-oncogenes, but they are in an inactivated state. These inactivated genes can be activated at certain conditions. Diagnostic methods of cancer: biopsy and histopathological studies. In biopsy, suspected tissue is cut into thin sections and is examined under microscope. So when we study this tissue under microscope, we can see the state, nature and structure of the cells in the tissue. If it is changed from normal state, then we can conclude that the tissue consists of cancer cells. The other techniques are radiography, CT scan, are techniques of diagnosing cancers. CT scan, 3D imaging of internal organs is generated by X-rays, and MRI scan, pathological and physiological changes in a living tissue are detected by using magnetic fields and non-ionizing radiations. Then immunological and molecular biological diagnostic method. These techniques can all be used to detect cancers. Identifying certain genes which make an individual susceptible to cancers can help to prevent cancers. Treatment of cancer. First one is radiotherapy. In radiotherapy, tumor cells are irradiated to death. Also, proper care is taken for protecting surrounding normal tissues during radiotherapy. Next one is chemotherapy or chemical therapy. Here, drugs specific for particular tumors are used to kill cancer cells. They have side effects such as hair loss or anemia. And immunotherapy, biological response or modifiers such as alpha interferons are used. They activate the immune system of patient and helps in destroying the tumor cells. Drug and addiction. Name of the drug is opioid. Source Papaver somniferum. Part of the plant used is latex. Mode of intake is snorting or injection. Effect binds with opioid receptors in the central nervous system and gastrointestinal tract. Depressant. It slows down body function. Example, diacetyl morphine and heroin can be called as smack. Next, cannabinoid. Its source is cannabis sativa. Part of the plant used is inflorescence, flower top, resins, leaves. Mode of intake inhalation or oral. It affects cardiovascular system. It binds with receptors in the brain. Example, marijuana and hashish. Then coca alkaloid. Erythroxylum coca is the source of this coca alkaloid. Part of the plant used is leaf. Mode of intake snorting. Effect, it stimulates central nervous system and gives sense of euphoria or increased energy. Interferes with transport of neurotransmitter called as dopamine. Excess intake will cause hallucination. Example for coca alkaloid is coke or crack. Then barbiturates, amphetamines, benzodiazepine and morphine. Effect, it cope up with mental illness like depression and insomnia. Nicotine or alkaloid. Source is tobacco. Part of the plant used leaves. Mode of intake is smoking or chewing. Effect, it stimulates adrenal gland to release adrenaline and noradrenaline. It raises blood pressure and heart rate, and it increases the risk of cancer of throat, lung, etc. And it can cause bronchitis, emphysema and CHD, chronic heart disease. And due to carbon monoxide mixed with blood, the carbon monoxide content in the blood increases and this reduces heme bound oxygen. Oxygen will not bind to hemoglobin due to carbon monoxide presence in the blood, and they become addictive to nicotine and alkali. The last one is LSD. Source of LSD is fungi and its effect is hallucination. Addiction and dependence. Addiction is a physiological attachment by a person to a certain effect of drugs such as euphoria and a temporary feeling of well-being, and these effects are associated with drugs and alcohol. If regular dose of drugs or alcohol is abruptly discontinued, or if drug or alcohol is stopped suddenly, that will show the following symptoms: anxiety, shakiness, nausea and sweating. Or again, if the drug is given, this anxiety, shakiness, nausea and sweating will be stopped. Prevention and control of drug abuse. Avoid undue peer pressure, which means friends pressure. Education and counseling can be given. Seeking help from parents and peers. Looking for danger signs. Seeking professional and medical help.